Welcome to the video series on fungal infections. These videos are designed to be carefully watched at your own pace prior to class so that you will be well prepared for an interactive in-class activity. This video series is comprised of 6 parts. This video is 27.1 and we have a busy agenda, so let's get started. In this first video we will establish a solid foundation of mycology, including fungal cell structure, fungal cell morphology, and common fungal cell vocabulary terms. We will also introduce you to the classes of fungal infections and their basic patterns, as well as the major classes of antifungals. Here we go. Fungi are enormously varied eukaryotes. There are 200,000 genera of fungi. Only 200 have been documented to cause human disease. But the vast majority of infections are caused by 10 or so genera of fungi, and we will focus our efforts on these. Fungi are defined by their ability to metabolize decaying organic matter, a property known as heterotrophy. Beyond this basic metabolic principle, however, fungi are remarkably varied. Generally speaking, they're almost always aerobic. They can reproduce sexually or asexually, and they can be unicellular or multicellular. We're going to have more on these last 2 points in a moment. Let's talk about fungal cell structure. Fungi have typical eukaryotic features, including a membrane-bound nucleus, a nucleolus, linear chromosomes, robustly active cytoskeleton, and many other membrane-bound organelles. Like many bacteria, fungi also have a rigid cell wall external to the cytoplasmic membrane. The chemical makeup of both the cell membrane and cell wall, however, is unique. As most of you remember, within the cell membrane many sterols are embedded that are paramount for physiologic cell function. In mammalian cells, the predominant sterol is cholesterol. In fungi, however, the predominant sterol is ergosterol, and this is extremely important. Ergosterol is a major target of pharmacologic intervention. It is synthesized from squalene, and there are 2 classes of medications that block the ergosterol synthesis pathway. The most commonly used is the azoles. These block lanosterol 14-alpha demethylase. The azoles are a commonly used class of fungistatic drugs with many different agents that each vary based on avidity for the enzyme, pharmacokinetic profile, and side effects. The second important class of drugs that blocks this pathway is known as the alloamines, which act by inhibiting squalene epoxidase in the early stages of ergosterol synthesis. A third class, the oldest and broadest spectrum agent of all the antifungals, is known as the polyenes. Polyenes bind to ergosterol already within the cell membrane. They form pores that allow cations and small molecules to leak out of the cell, resulting in fungal cell death. In addition to ergosterol, however, polyenes also bind to many other sterols in human cells, including cholesterol. This nonspecific binding of sterols in human cells results in a considerable amount of toxicity. Now let's step back from our discussion about the cell membrane of fungi and talk a little bit about the cell wall. The cell wall of fungi is markedly different from bacteria cells. It's made up primarily of the polysaccharides mannan, which forms a large manoprotein on the outside shell of the cell wall, glucan, and chitin. An enzyme called 1,3-beta-glucan synthase is one of the major synthesizers of glucan for the cell wall, and it's a special target for pharmacological intervention. A class of antifungal medications known as echinocandins are the class of medication that inhibit this enzyme. Now please note that this was not meant to be an in-depth look at fungal cell structure, but rather a look at what unique aspects of fungal cell structure drive pharmacological intervention. Let's step back and discuss fungal cell morphology while introducing some really key vocabulary terms. As we said earlier, fungi may reproduce from asexual or sexual processes. Asexual reproduction yields a daughter cell, known as a conidia. Sexual reproduction yields a daughter cell known as a spore. There are 2 major morphologies of fungi, yeasts and molds. A yeast is the simplest to understand. It's usually just a small round cell. When it reproduces, it generates a bud, which is really just a round outpocketing. Molds, on the other hand, are quite differently shaped. Molds are made up of tube-like extensions with parallel walls known as hyphae. The hyphae are usually, but not always, divided into subunits by septa, which themselves can have one or many pores. These hyphae can become an intertwined mass, which is often referred to as a mycelium. The mycelium behaves like a root, growing into the decaying material. Notably, some yeast species, like Candida, can also grow structures that resemble hyphae. These well-named structures are known as pseudohyphae, and they're simply tubular buds from a yeast that look very much like the hyphae that grow from molds. However, the wall of pseudohyphae are usually much more fragile than the wall of true hyphae from molds. While most fungi grow exclusively as a yeast or exclusively as a mold, some species can grow in either phase. The phase in which the species exists is often dependent on environmental clues. When their environmental reservoir is at ambient temperatures, meaning less than 37 degrees, they tend to grow as molds. Molds tend to be more metabolically active. Once the temperature goes above 37 degrees, however, it's a sign that environmental conditions are a little more harsh, and so it's time to expend a little less energy. In these conditions, the dimorphic species convert into their yeast forms. This conversion is what is known as a heat shock response. In addition to understanding the metabolic changes associated with a heat shock response, you should also note that molds are typically more immunogenic than yeasts. So it makes sense then that these organisms convert into their yeast form when inside the human body. Yeasts are more likely to avoid an immunological response from the host. The morphological shapes of yeasts, molds, conidia, spores, and mycelium are all quite variable. It makes sense then that the gold standard for diagnosing all fungal infections is direct microscopic observation. Fungal cultures are commonly performed, but they are often very slow and require selective media. Many serologic and urinary antigen tests are available, but while some are quite sensitive, they often lack specificity. We will discuss the diagnostic test of choice for each disease entity as we come across it. Traditionally, fungal infections are classified either based on their anatomic location, meaning are they superficial, mucocutaneous, or do they infect the deeper organ structures? Or they're classified based on their epidemiology. Are they opportunistic? They tend to infect immuno-incompetent individuals only. Or are they endemic, meaning they could infect almost anyone? As each type of infection is discussed, remember to pay special focus to fungal morphology. Are we dealing with a yeast, a mold, or one of those clever dimorphic organisms? By keeping these distinctions clear in your mind, the diseases themselves will be easier to understand and easier to remember. The first anatomical classification of fungal infections we mentioned was superficial fungal infections. This means that the infection is limited to the cutaneous and subcutaneous tissues. Within this group, we will discuss a cutaneous mold called dermatophytosis, a cutaneous yeast causing a disease known as tinea versicolor, and one cutaneous dimorphic organism, sporotrichosis. With superficial infections, inoculation occurs most commonly through direct environmental exposure of the skin and subcutaneous tissues to the pathogen. The next 3 videos in the series will focus on epidemiological distinctions of infections, specifically the endemic mycoses and the opportunistic mycoses. Our discussion of the endemic mycoses will focus on 3 fungi, each a group of dimorphic organisms. These fungi are not part of a normal microflora. It's very helpful to understand the basic pattern that results from this group of organisms. Now, think back to TB here, because this group of infections behaves similarly to tuberculosis. First, there's inhalation of environmental inoculus. In this case, the dimorphic species Conidia. Once inhaled into the lung parenchyma, the dimorph causes a granulomatous response as the body attempts to prevent systemic infection. Usually, this works, and the host is either asymptomatic or has some pulmonary symptomatology. As with TB, however, a particularly large or virulent inoculate a host genetically predisposed to infection, or some degree of acquired host immunocompetency results in more systemic spread or reactivation of the latent infection. Opportunistic fungal infections are covered in the fourth video, 27.4. These fungi are ubiquitously found in the human flora and usually only cause disease in an immunocompromised host, such as a transplant or HIV patient. In this fourth video, we will discuss 3 common opportunistic fungi, 1 yeast and 2 molds. Candida, an opportunistic yeast, and the most common of all of these fungal infections, will be discussed separately in the 5th video due to its slightly increased complexity of presentation. Despite these classifications, please know that some fungal infections do not obey these rules as I've laid them out for you. Our primary response to fungi is neutrophil-mediated killing. Fungi that escape neutrophil destruction are then slowed, but often not killed, by macrophages. Macrophages activate a subsequent Th1 predominant response that appears to be instrumental in preventing systemic spread of infection. A humoral antibody response may be detected during the course of most fungal infections, but it appears to play a varying, although usually limited, role in controlling the infection itself. That's it for the first video. Believe it or not, we've covered a lot of material. Now take a moment to review these slides, which, while very basic, illustrate the major take-home points of the lecture. See you next time.